it's probably important to just tell you what the question was that the research was trying to answer, because one always should start with a question before you try and say anything at all. And the question was whether the different cells of the body have the same genes or they don't. And at the time of this work, that was an entirely open question. And indeed, in the 1800s, it had been indeed suggested that cells might not have the same genes. The conclusion was that different kinds of cells of the body do all have the same set of genes.
What do we mean when we talk about epigenetics? The caterpillar and the butterfly share exactly the same DNA sequence, but they look very different. And this is what we mean by a change in cellular phenotype. Now, we also know that to make that change happen, there are lots of genes being switched on and off. So that fulfills the definition of a change in cellular phenotype, which also involves changes in gene expression. But those two forms of the animal have completely identical DNA sequence. So this is a change in gene expression and cellular phenotype without a change in the underlying DNA sequence. But what makes it epigenetics is this word, the mechanism.
EpigeneSys, if it has to be defined in a nutshell, it's a network of excellence that has been funded by the European funding system in the context of the 7th framework. The objective is to bring together a series of groups throughout Europe to try and tackle an important question that can be really changing paradigm in the approach.
When the Network of Excellence EpigeneSys was created, there was a realization that the field of epigenetics was really starting to come to fruition, in the sense that researchers were starting to make discoveries about how gene expression states are inherited and how this can be influenced, for example, in diseases such as cancer, and also how these stable gene expression states are reprogrammed.
The point of debate is mainly on the transmission from generation to generation. So transmission from one cell to the next is something that many people really like to understand, how you have information that can be passed from one cell to the next. Then what is a lot more challenging is to think about how to make this transmission going at the level of the organism, germline, so from one parent to the children and on over.
There are a lot of very good epigeneticists in Europe, and we felt that it was important that we remain competitive and also we remain at the forefront by linking up with systems biologists. That was the beginning of EpigeneSys, a network that would bring together epigeneticists and systems biologists with a view to making a new era of research that would involve the systems biology of epigenetics.
The goal was really to achieve this capacity of blending different disciplines, people that were more in the biological question and people that had the tools from the mathematical perspective, but also in terms of the physics of the nucleus and its organization.
But EpigeneSys was born out of a network called Epigenome that many of us set up 10 years ago. And at the end of that network, we realized that a lot of the data we began to generate was becoming very data intense, big data, if you like to think of it that way. And it needed to have experts in modeling and in computational approaches to try and understand it and to use statistical power to figure out whether we were being biased in the conclusions that we were drawing. So this has been a collaboration between computational scientists and more cell biology and molecular-based scientists.
One of the great achievements of the EpigeneSys network, I believe, was the fact that right from the start, we wanted to bring in some of the younger researchers, the new rising talents both in the field of epigenetics and in systems biology.
I am a member, a RISE 1 member of EpigeneSys. That basically means that I was selected as a young group leader when I started my lab. Probably one of the most important things that I have got through EpigeneSys is, on one hand, the possibility to start new collaborations with very good people doing epigenetics and systems biology, which is something that I don't do myself, so it has been a great complementarity.
And I joined the EpigeneSys Network of Excellence 5 years ago. I think the most important moment was when I was actually elected as a RISE 1 member, because I got introduced to the European community. And this is where I realized all the opportunities I could have to develop my lab. I could put face on names from articles I was reading and talk to those people and get some very important feedback from my research.
I'm part of this consortia since very beginning. I did my PhD in Europe, my postdoc in Europe, and now I'm continuing on my lab. This consortia has been very important in many fundamental aspects. This allowed me a good platform to meet high-profile scientists and offer access to their laboratories, their networks, the workshops. They've been all outstanding.
So I work on bioinformatics, which means that I only analyze data. I do not work on the lab. And my main focus is on epigenetics linked to transgenerational inheritance. This means that I study how features acquired during the life of an organism get transmitted to the following generation.
It's very important for new PIs, it's very important for postdocs and PhD students. It's important also for people who are established already, because without a community, it's like being without roots. So you get detached and disconnected from the rest of the field.
I'm a PhD student in the lab of Robin Alshire in Edinburgh. And I mainly know EpigeneSys through the meetings that have been organized. I'd never really heard about it before. You hear a lot before you're a scientist about genetics, but never really about epigenetics. And I thought it was a fascinating extra layer that was completely unexplored. And I found out that Edinburgh was a great place to do it. And I joined the Wellcome Trust program, which let me sample different labs and do different things. And epigenetics was still what I wanted to do at the end of that. And I was very happy to join the lab in Edinburgh and continue to look into that.
So it's wonderful to be here in Paris for unfortunately the last ever EpigeneSys meeting. You will see many happy people here, but you also see many people that have half a tear in their eyes, because this is the very last meeting of this wonderfully successful program, EpigeneSys, which started about 5 years ago as kind of a group of people in Europe that were hugely excited about epigenetics, brought the young people into the network. This has created fantastic new opportunities for the training, for the dissemination of epigenetic knowledge in Europe.
EpigeneSys, the epigenetics community in Europe, definitely has an identity. Hopefully it'll be able to let that live and breathe on in the future.
The annual meeting, I consider this meeting to be the most special and important moment where really everybody can gather together and exchange ideas, present results, but not just present results, really discuss about the present and the future of this field. This is the most important thing, I think. Yeah.
Nothing beats getting people together and allowing them to exchange scientifically, whether in the form of formal presentations, scientific talks, or over a cup of coffee, a croissant, or at the bar. That's when the real science starts to get done, because that's when the connections can be made between two or more human minds, and ideas actually emerge from this kind of interaction?
At the very beginning, to be honest, it was not obvious that things could work, because we were trying to bring together people that were not used to talk to each other. So we had to find a kind of a common language. So it was like building up Babel. I can certainly remember the very first meeting when we were kicking off the network. We were just a few people, and we asked the question to the audience, how many people can understand how to do some mathematical modeling? And we barely had a couple of hands being raised. And we asked the same question coming at the end, and it was just amazing to see that now we could see a lot more people capable of doing it. And so they've learned from each other, but it's also because the network is by itself a system that has been very integrative.
We felt that one of the most important things for a network is to allow scientists in different laboratories to exchange at the level of the experiments. So rather than reinventing the wheel in every lab and not necessarily doing it with the right infrastructure, why not make the most of the existing equipment and technical know-how and actually allow the experiments to happen in the places where they're best placed to be carried out.
I started as a fly geneticist, but now I work both in flies and in human model.
I'm actually one of the founding members of EpigeneSys. I work on proteomics. I do histone quantitation of post-translational modifications, and we know each other for quite some time. And so now we meet, and through EpigeneSys, we actually met quite a few times. Yeah. And now we have this opportunity to do quantitative measurement using Curie's excellent method.
Absolutely, and in fact, just inspired by the last talk of Amos Tanay. So we would like to basically look at dynamics of epigenetic memory. That's the future, I think. What do you think?
I mean, it's funny that you mentioned Amos's talk, because I was one of the few that said, okay, so we do use, or at least we try to use, differential equations to actually find out something and to use it to predict something.
Right, and yeah, that will be quite testable.
I think that's EpigeneSys at its heart.
Let's do it!
I think the really amazing thing that EpigeneSys created was a team spirit in Europe. We are now a society, we are now a unit that brings together scientists from all over Europe for the benefit of basic research. And I think this is a really big achievement for European science.
And science is really important. One is always at the forefront, one hopes. One's asking questions that have no answers. So the network was tremendously important in allowing this nurturing environment that would provide new answers to existing questions.
One aspect into which EpigeneSys has also taken important initiatives was in terms of discussing with artists and thinking about how some aspect of the science was carried out in the network could be brought to the public.
We've tried to find a new way of addressing people and involving people and engaging them. So we've decided to marry science and art.
One of the first things that interested me was the work of C.H. Waddington, and particularly his model of the epigenetic landscape. So a lot of the work that I've been doing has been remodeling or revisiting this idea of the epigenetic landscape.
For me, the future should be about more networking and more such networks. And I think that's one of the wonderful things about science. It's international, it has no frontiers, or it shouldn't have. And being able to be part of this network, in some ways, was really what science, I feel, should be about. A sort of dream come true, a dream that's a reality, which is that all over the world, scientists have the same objectives, which is to try and understand how, in the case of biology, how life works, and to ask some very simple questions and try and get answers from them through exchanges.
The idea indeed was to try to erase borders between disciplines. And I think the time we are now is really ripe for a change in terms of the way we work. But I think where we have really achieved our goals was to try to bring together a community and make a difference. So this, I think, is something we can be very proud of, and there's more to do later. However, the EU has decided at this point in time not to continue this scheme.
So the future of epigenesis is bright. Although the 5 years are ending, I think the community has come together in such a strong manner that we will continue in many forms. It's actually a new beginning for looking into other aspects of how we can foster epigenetic research in Europe.
I think that we can really be very, very happy and pleased to have contributed with many important discoveries in terms of understanding principle of organization of chromatin in the nucleus, for example. But this is only the beginning, and clearly there's much more to be done, and a lot of other countries, like in the US or Japan and China, are trying to take things further. So it would be a pity if in Europe we were not in a position to capitalize on what we were first to put forward. But at the same time, I wouldn't like to convey a wrong message and pretend that we are just about to solve big things and that we will cure in a flash many diseases that are existing. That I think would be totally unrealistic, and the public is to be respected and well informed. And I think it is essential to realize that there is time that is needed. And even if we try as much as we can, and by setting up organization and working in a collaborative way as we have been able to do it, we can make things happen much faster. There is still a need to take some time to do things in a proper way and achieve unexpected goals as well.